In this lecture we'll cover infectious diseases of the nervous system, focusing mostly on meningitis and encephalitis. These are more common than the other types of infections such as prion diseases or brain-eating amoebas, but we'll touch on those at the end. First, we'll talk more about the inflammatory based diseases like meningitis and encephalitis. Meningitis, of course, affects the meninges, while encephalitis affects the brain tissue. That's the encephal portion of there, so it's in the brain rather than the meninges. The slide over here is showing us on the left brain tissue with a whole bunch of holes in it, and the brain tissue on the right is fairly normal. The reason that we have a whole bunch of holes in that brain tissue on the left is because of an infection. We'll touch on this again at the end. This is what we call transmissible spongiform encephalopathy. That's what prions cause. But first, let's talk about more common infections that would be viral and bacterial infections. Viral infections being a whole lot more common than bacterial. When we have infection in the meninges or within the brain tissue, this leads to an inflammatory response, and that is an issue for enclosed structures like the brain. We're going to have to worry about increases in intracranial pressure. If it's a bacterial or fungal infection, we also have to worry a bit about the pathogen reproducing and killing the nearby neurons. For some viruses, we also have to again worry about cell death. So a couple things are a problem here. Direct destruction of nervous tissue by the pathogen, and then an increase in intracranial pressure. That one's not new. Alright, let's review inflammation a little bit. So, the whole purpose of inflammation is to clean up some kind of injury. Now, this could be actually an injury. It could be cell death, for example. So if a cell ruptures open, well, the contents that were inside are now outside. There's no difference between inside and out anymore. So this will release what we call damage-associated molecular patterns, DAMPs. This is just a nice way of saying very small things that are associated with damage. So the molecules that leak out of damaged cells. This could be from the nucleus, the mitochondria, the cytoplasm, doesn't matter. Something that used to be inside is now outside. Alternatively, something that was never inside of us now is. We might have ourselves some sort of infectious pathogen. Well, the surface proteins on that pathogen are going to be recognized as a pathogen-associated molecular pattern. PAMPs and DAMPs. Stuff associated with pathogens or damage. And in either case, we need some inflammation to clean it up. That's the whole purpose of inflammation, whether you have an infection or damage like we can see here. So on top, normal heart muscle, on the bottom, heart muscle following myocardial infarct. So, damage to the heart muscle led to an inflammatory response, and you'll see a whole bunch of little purple dots over there. Those are white blood cells invading the site of damage. That's what it's all about. If we find these, either damage or infection, we need to increase blood flow. So we're going to cause vasodilation, and we're going to increase the permeability of our blood vessels so we can pull out white blood cells to clean up the mess. Those PAMPs and DAMPs will be detected by immune cells, of course, using surface proteins. Everything that happens in life is done by a protein. So my white blood cell here, or in the case of the central nervous system, maybe a microglia, whatever the macrophage is here, has a variety of receptors to pick these up. There's a big class of them called Toll-like receptors. When DAMPs or PAMPs bind to Toll-like receptors. This then triggers an inflammatory response. And it's going to do it kind of in two ways. Kind of the slower process that's going to act over a longer time course involves changing gene expression. So we're going to have to stimulate some transcription factor, in this case NF-kappa beta. You know, I got a whole whiteboard here. Why am I not using it? Like I got to save space for something. Okay, so we activate a transcription factor. This one has a name, NF-kappa beta. And it does what every other transcription factor does. It goes to the nucleus and affects gene expression. In this case, we're going to increase the expression of pro-inflammatory cytokines. All right, so we'll make some RNA here, and that'll get translated, what's wrong with me, into pro-inflammatory cytokines. And the one that we're talking about in a little more detail is TNF-alpha. This is not the active form, so we call it pro-TNF-alpha. Pro meaning before. So it's not quite mature yet. The second thing that Toll-like receptors do are activate a collection of enzymes that we call shedases. Very nice name, that's what they do. They shed surface proteins. And I always like to draw these as a pair of scissors. So these are going to cleave my pro-TNF-alpha. That's what my shedase is going to do here. This one has a few names, Atom 17. I'm going to go ahead and list it because we'll see it on another slide. So my shedase cuts this pro-TNF-alpha, liberating the active form TNF-alpha, tumor necrosis factor alpha. Tumor necrosis factor then triggers vasodilation by acting through epithelial cells. So the macrophages create TNF-alpha, and what does that TNF-alpha do? I think there's some data for us to look at here first. Okay, these data are showing us that Atom 17, that shedase, is critical for releasing TNF-alpha. So the bars there are showing us the amount of TNF-alpha that was released, and then on the x-axis they're showing us a few different PAMPs, if you will. LPS, lipopolysaccharide, that is an isolated PAMP. So that's a component of the bacterial cell wall. They also just added some different bacteria, which of course are covered with PAMPs. So the isolated PAMP does a little better job because it's just a solution of free-floating LPS. Anywho, you put on some PAMPs, you get a whole lot of tumor necrosis factor unless you knock out Atom 17. And the striped bars that you can't see because the level of TNF-alpha is basically 0, those are from Atom 17 knockouts. So if you don't have your shedase, you don't cut pro-TNF-alpha, you don't liberate TNF-alpha. That's all that's showing us. Okay, so what does TNF-alpha do for us? So we'll leave that one floating around. And now we're going to think about some kind of epithelial cell here. And these have tumor necrosis factor receptors. They're the receptors for tumor necrosis factor, so the name works out in this case. When I activate my tumor necrosis factor receptor, this triggers the expression of cyclooxygenase, which then makes prostaglandins. So I'm just going to kind of brush all that under the rug and say we create prostaglandins. For example, prostaglandin E2. And that's what these data are showing us. So by applying different doses of TNF-alpha, we can see the expression of cyclooxygenase, that's shown in panel C. Cyclooxygenase, of course, is an enzyme that's going to create prostaglandins. So the main thing that matters here, I don't think you care about the time course, look at panel F, the bottom right. Prostaglandin release from epithelial cells is triggered by TNF-alpha. prostaglandins then act on our smooth muscles, so vascular smooth muscle cell, there it is. When these things when they contract, we get vasoconstriction. When they relax, we get vasodilation. And prostaglandin E2 causes vasodilation. So when we have DAMPs and PAMPs, those act on Toll-like receptors to cause the release of TNF-alpha from the macrophages. TNF-alpha then triggers the release of prostaglandins from epithelial cells. Prostaglandins then cause vasodilation of the vascular smooth muscle cells, causing an increase in blood flow at the site where we have DAMPs and PAMPs, so that we can increase the amount of white blood cells that are going to come by, and we also increase the permeability of blood vessels, so it's easier for white blood cells to come out of the blood vessel and into the site of damage or infection. So there's inflammation in a nutshell. This is a wonderful thing unless you're encased in bone and you have a finite volume. So if you get a boo-boo out here in the periphery, you'll get a little bit of swelling. It'll turn red, no big deal. Kind of a big deal for the brain and meninges though, because the skull creates a finite volume. And when you're pulling out more cells and fluid, well, they occupy space. And there's only so much space in the head. All right, let's start with a little review of the meninges, just so we all remember what these are. Alright, so we got some scalp and hair and whatnot. No one really cares about that right now. Below that we've got a skull. Okay, so we don't really care about the skull yet. I mean, this matters because it creates a finite volume. Now, below the skull, there's where we have our dura. And the dura has two layers. There's the periosteal layer that hugs the skull a whole lot closer than I've drawn it here. And then there's the meningeal layer that actually hugs the brain tissue. We'll get our brain up here in just a little bit. Under the dura, we have the arachnoid. And the arachnoid layer is filled with cerebrospinal fluid, because it contains these arachnoid processes there that create an open space called the subarachnoid space. So this right here is filled with cerebrospinal fluid. And it also has a whole lot of blood vessels running in it. So this will be a common site for pathogens to enter. It's in that subarachnoid space. Below there we have the pia mater, and I've said this before, I'll say it again. The pia mater ain't shit. It'll fall apart if you look at it wrong. It's not really there to provide structure, it's more there to be a permeable, delicate covering of the brain to ensure that it stays moist. Doesn't really seem to do a whole lot for us. And then finally we get into brain tissue. But the first thing that we encounter once we get into the brain, we'll make our brain purple. The first thing we have is a whole bunch of glia, in particular astrocytes, that form a barrier that we call the glial limitans. So the closest thing to the meninges is of course not neurons. They're delicate. What we put out there are the glia. Oh, yeah, I know I said the pia isn't really that important, but I might as well label it. It doesn't deserve much of a label, but we'll give it one. So under the skull we got the dura. Under the dura we got the arachnoid, and the arachnoid has that CSF-filled subarachnoid space, which also has blood vessels running through it. Under the arachnoid is the pia. Then we're finally in the brain. The first thing we encounter, glia. Those things are not postmitotic. We can make more of them. They're a much better barrier to have than neurons. So here's one way of looking at it. Here's another way of looking at the meninges, if you like, a little more cartoon. There it is. All right, let's get into meningitis then. So meningitis can be caused by bacterial infection or viral infection, and viral infections are a whole lot more common. Viruses are smaller. Viruses are also pretty good at getting into places. So viral meningitis is a whole lot more common than bacterial, and that's good because it has a much better prognosis. Alrighty, as far as risk factors go, well, because we're dealing with viral meningitis most commonly, one of the biggest risk factors is not being vaccinated against those pathogens. So there's a reason why we have common vaccination as we enter our first year of life and move on. We get a whole bunch of vaccines. In order to go to school, you got to get your vaccines. Because schools are a common place of spreading disease, and some of these diseases can affect the nervous system. And although they might have a good prognosis, it doesn't mean you always escape unscathed. So vaccination is very useful. Here we can see the drop off in cases here. What we're looking at are the number of cases of meningitis. And following vaccination, which started in 2000-2001 there, we see that the rates of meningitis dropped dramatically to almost nothing. So, having vaccination against neurotropic viruses, mumps, measles, rubella, that's a very common vaccine that everyone gets, certainly decreases your risk. So if you're one of those unvaccinated folks, well, you're probably not unvaccinated for those because you got them when you were a kid, but you're at risk by avoiding vaccines. Also, exposure to neurotropic pathogens, that's a big risk factor as well. So exposure to any virus or bacterium that can get into the brain there, that's going to increase your risk. If you have compromised immune function, that certainly increases your risk because you're not properly identifying and destroying pathogens before they have a chance to settle in the central nervous system. And then, of course, those who are very young or very old. Very old is a risk factor because you are more likely to have damage in your nervous system. Could be a neurodegenerative disorder or a tumor, whatever it may be. Your blood-brain barrier has a greater chance of not being intact in older age because you've had damage. Young age is a big risk factor because the blood-brain barrier hasn't developed yet. So that's what it's all about, having that blood-brain barrier intact so that things in the blood can't exit and get into the brain. The blood-brain barrier develops over time. All right, when we're talking blood-brain barrier, let's say this is a blood vessel. Okay, there's some smooth muscle around it, but I don't care about that for now. In the brain, there is no space between our vascular epithelial cells. They're connected by what we call tight junctions. Nothing can pass through. No going between the epithelial cells. So all of our happy white blood cells stay in there, and any of the angry pathogens that might be floating around stay in there. But in the very young, where we haven't yet developed these tight junctions, well, we got a space for pathogens to exit. And like I said, in the elderly, in case you have any sort of brain damage, or neurodegenerative states, blood-brain barrier doesn't work quite as well. So the biggest risk factors can be really summarized as lack of vaccination and then any condition that compromises blood-brain barrier function. Now as far as symptoms go, well, classically we're going to see neck stiffness and headache and of course fever because you have an infection, either bacterial or viral. So those are the three common symptoms for meningitis. Because we have infection in the meninges and they get irritated, that causes headache by directly stimulating either the 5th or 10th cranial nerve, depending on where we're talking about, whether it's the forebrain or the brain stem. That infection of the meninges causes headache. And of course, if it goes a little lower, we're going to feel that as neck ache and neck stiffness. The infection leads to inflammation, which can lead to intracranial pressure buildup. So elevated intracranial pressure is another common symptom of meningitis. That elevated intracranial pressure, of course, causes your typical signs of nausea. It can affect cranial nerve function. It can certainly lead to seizure activity. It can alter consciousness. If bad enough, of course, it can lead to coma and death. As far as diagnosis goes, taking a CSF sample is the gold standard. If we want to say you have an infection in your central nervous system, we're going to pull out a little bit of CSF. We want to see if anything nasty got out of the blood and into the brain or meninges. Well, if it did, we'll detect it with a CSF sample. So right now we're kind of all about the meninges. Those are of course going to wrap around the brain, brain stem, spinal cord. Don't worry, I don't have any visual disturbances. I ran this down for a reason. In here we have our cauda equina. We have our lumbar cistern. That's why we'll do a lumbar puncture to grab some CSF. Of course, assuming we don't have any elevated intracranial pressure. If we do, best not to do a lumbar puncture. CSF is going to spray out. But down in this area where we don't have spinal cord, we also have very poor innervation of the dura. We can jab a needle in there, pull out some CSF, and get it tested. Do we see bacteria in our CSF? If so, you got yourself a bacterial infection. On the other hand, do we see no bacteria, but still a whole bunch of white blood cells? We should see basically none. There should be almost no white blood cells in our CSF, because we should have a blood-brain barrier keeping all the white blood cells out. So if we find a whole lot of white blood cells, but no bacteria, that's viral meningitis in that case. Of course, this needs to accompany the classical signs, so we'd expect to see signs of meningitis as well. That's why we would do the lumbar puncture. We're not just doing it for fun. Now, MRI can be very useful in distinguishing whether it's meningitis or encephalitis, because they will have very similar symptoms. So how can we tell them apart? Well, have a look over here. On the left, we got T1 MRI. On the right, T1 MRI with a contrast agent. The contrast agent shows us where we have blood-brain barrier permeabilization. So let's look on the bottom. It shows us a nice parasagittal view of the brainstem, spinal cord. You'll notice when we look at from left to right, so without contrast and with, the meninges light up with that contrast agent, showing us we have permeabilization of the blood-brain barrier within the meninges. Clearly we're dealing with meningitis. we'll see an example of encephalitis in a bit whenever we get to it. So your MRI imaging is useful for sure, but as far as saying that you most certainly have bacterial or viral infection, CSF samples. That's what we want to take there. Now, as far as pathophysiology goes, whether it's viral or bacterial doesn't really matter. Once they enter into the CSF, most commonly in this subarachnoid space, now we have a whole bunch of PAMPs. So any of the glia or even the epithelial cells are going to pick these up. The PAMPs, of course, trigger TNF-alpha release. TNF-alpha triggers prostaglandin release, so we get vasodilation, increasing blood flow. We're pulling out fluid from these now permeabilized blood vessels. So fluid and white blood cell come out. Well, they occupy space, so we're going to get some swelling, right? And that swelling, right, the inflammation-dependent edema that occurs, elevates intracranial pressure. So we'll get those common signs there. If bad enough, brain tissue can herniate as a result. So if we're swelling up in the meninges, that can push brain tissue over, and that brain herniation can, of course, obstruct blood flow. It can obstruct the flow of CSF. Both of these are going to cause downstream damage to neurons as well. The pressure itself can also just mechanically stimulate neurons. Remember, everything's mechanically gated to some degree. So if you rub on your eyes, you see colors. If you push on a neuron, you're going to force open ion channels and activate that neuron. So this could lead to excitotoxicity. So lots of potential bad things. If we herniate that brain tissue over and obstruct CSF flow, for example, we'll see hydrocephalus, as we can see on the bottom. We'll see swelling of the ventricles, further increasing intracranial pressure. So the rise in intracranial pressure is a big problem that needs to be controlled. As far as the neurological symptoms, well, that depends. Is it more forebrain, or is it more brainstem that's being affected? If it's in the forebrain, we're going to get some signs of cortical abnormalities, so you might see some very kooky changes in cognitive function. You might see sensory motor changes. If it's in the brainstem, you could still get sensory motor changes. Here we're also going to affect cranial nerve function. And of course the brainstem carries out vital functions, so we might have problems with respiration. So it really, of course, depends on location. As far as what makes meningitis unique, it's really there's signs of infection, and then you're getting these headaches and signs of elevated intracranial pressure. As far as treatment goes, well, for viral meningitis, not much is needed. If there's an increase in intracranial pressure, control it. But viral meningitis often resolves all on its own with very little ill effects. It's usually not fatal, I can say that. It's almost always not fatal. There may be some long-lived sensory motor or cognitive deficits, depending on how pronounced the intracranial pressure increases were. Bacterial meningitis fares a whole lot worse than viral meningitis. So this will require treatment. We need antibiotics. Bacterial meningitis has a much worse prognosis, even with proper control. Neurological complications are going to occur in most cases of bacterial meningitis, and it can be fatal, of course, depending on who we're talking about, 5 to 25% of the time. Now, as far as controlling intracranial pressure, we do the same thing we would anywhere else. You can use surgical decompression, so you can open up a hole, let the brain kind of herniate out that hole to relieve the pressure. You can use mannitol to dehydrate the brain, so pull excess fluid out. You can also use steroids. Steroids you wouldn't use with traumatic brain injury, but in this case you would. They're safe to use for meningitis and encephalitis. And what corticosteroids do is really the opposite of TNF-alpha. And so this cartoon is showing us that here. So the big red nasty looking box, that's TNF-alpha and other pro-inflammatory cytokines like interleukins. So these bind to their receptors, turn on that transcription factor, NF-kappa beta. There it is again. That's why I named it, just so this would make sense a little bit later. And that transcription factor causes the expression of pro-inflammatory cytokines. Corticosteroids, on the other hand, act on their own receptor, glucocorticoid receptors, for example. These are also transcription factors, but rather than turning on pro-inflammatory cytokines, we turn them off. So we reduce the expression of those pro-inflammatory genes. By cutting down on inflammation, we of course decrease the amount of swelling that occurs and thus lower intracranial pressure. If seizures occur, anticonvulsants are used just like they were in past lectures. So again, we're going to inhibit voltage-gated sodium channels, voltage-gated calcium channels, or GABA transaminase. As far as prognosis goes, pretty good for viral meningitis, not so good for bacterial. Here we can see how the mortality of bacterial meningitis varies across age. Again, the very young don't do quite as well. They don't have that blood-brain barrier intact yet. Once you get into early and later childhood, that's when you kind of have your best fitness. It's kind of hard to kill them in that age range. They got their blood-brain barrier intact and they're young enough that recovery is pretty good. As you get older, so as you enter early adulthood and especially later adulthood, well then the mortality rates rise. Encephalitis does not affect the meninges, but instead the brain tissue. So we're in the brain parenchyma in this case, not the meninges. So on top, that's our good old meningitis. Right? You can see the meninges light up with contrast. The bottom is showing us encephalitis. Notice what's lighting up here, not the meninges, but the brain tissue itself. Down there in the, looks like temporal lobe, so it's probably some kind of herpes encephalitis. Great news is risk factors, symptoms, diagnosis, it's all the same as meningitis. We're going to use MRI to distinguish encephalitis and meningitis, but you'll see similar things. Like meningitis, more commonly we're dealing with viral encephalitis rather than bacterial. So bacterial and fungal infections are pretty darn rare to make it into the brain tissue. It's almost always viral infection for encephalitis. But the good thing is risk factors, symptoms, diagnosis, everything is the same. We're just in a slightly different location. Rather than meninges, we've gone a little bit deeper and we're in the brain tissue. Pathophysiology, really the same. We have swelling. That swelling is not good, right? We get an increase in intracranial pressure. All that stuff is exactly the same. Now, how do we get into the brain tissue itself? Well, it could be crossing from a blood vessel that's in the brain rather than the subarachnoid space. But viruses also have an additional option to get into nervous tissue, and that's by retrograde transport. Hopefully you remember retrograde transport. This is how those neurotrophin receptors make it back to the cell body so they can keep us alive. So neurons then, kind of by design, move stuff back up their axon. They have to. So retrograde transport is a wonderful thing, except when it's not. It's wonderful whenever we're trafficking neurotrophin receptors back to the cell body. It's not so wonderful whenever we've pulled in one of those surface proteins that had a virus attached to it. Now we're pulling the virus back into the cell body. And if this is something like a motor neuron, well, that of course lives in the anterior horn of my spinal cord, and it sends its axon out into the periphery. I get bitten by a dog that has rabies. Rabies virus is going to attach to the axons of motor neurons and get carried back into the central nervous system. No need to even worry about that blood-brain barrier. We're going to ride along inside of axons. There are several viruses that are going to enter through this route. So we can enter through the nose, infecting the olfactory nerve. We're not very far from the frontal lobes at that point. We could also enter through sensory or motor neurons because of peripheral infection. So in this case, we might not even need to worry about that blood-brain barrier. We're going to hop on the microtubule express and use the retrograde transport that goes on constantly within the axon to bring us into the central nervous system. Now as far as treatment goes, same as meningitis, right? Nothing new here. We still want to cut down on the swelling. We still want to control any of those dangerous rises in intracranial pressure that occur. If they're seizures, we control them the same way that we do in every other lecture. If available, antivirals can be useful for controlling the spread of the virus once we determine that we have viral encephalitis. So antivirals are going to be a little more helpful here. Unlike viral meningitis, which is largely going to resolve spontaneously, viral encephalitis usually has poorer prognosis than viral meningitis. Certainly, some viruses aren't as bad, right? Mumps, okay, usually doesn't cause encephalitis. And when it does, usually not fatal. That's not what you'd like to hear about any condition that might affect you. But certainly not as bad as herpes simplex encephalitis. Herpes encephalitis is going to cause death in 10 to 20% of cases, and in all the rest of the cases it's going to cause profound memory and language impairments. Herpes simplex encephalitis is awful. Also, like meningitis, vaccination is still wonderful. Here's a couple other examples of how the introduction of the mumps vaccine dramatically cut down on mumps rates. The top there, you'll notice the y-axis has a standard linear scale, so you see the dramatic drop-off. The bottom scale is on a log scale. So even though it looks like a straight line, no. Notice on that y-axis we're rapidly approaching 0. So it really does look like this again, but we've just changed the y-axis. Anywho, there are certainly other infectious agents that can infect the nervous system and destroy neurons. Right now, viruses, in addition to causing that increase in intracranial pressure, which is not a good thing, they can also directly destroy neurons. This is another way that viruses can cause damage. So certainly that swelling that occurs is not a good thing, but viruses reproduce usually by killing off the host cell. So these are not good house guests. They don't go out the front door, they just blow up the house, and that's how they leave. So viral replication can occur without killing the host cell, but it's often going to occur in a disastrous manner where it causes rupture of the host cell. That's not a good thing. So viruses, of course, aren't really alive. We won't get into that debate here, but they certainly aren't cells. They can't make their own proteins. They can't really do anything for themselves. So what they do is infect a host cell and use its transcriptional and translational machinery to make more viral RNA and proteins. If it has a DNA-based genome, it'll use the host DNA polymerase to copy itself. But it needs a host cell. After the host cell has done its job of making enough of the virus, cell ruptures, releasing the virus. So clearly not a good thing, because neurons are irreplaceable. So any viruses that can affect neurons will eventually kill them off. For example, West Nile virus or polio virus are both going to target motor neurons. West Nile virus will get a few other types of neurons, so you might have some cognitive changes with West Nile virus infection, but with both West Nile virus and polio virus infection, the destruction of motor neurons can lead to flaccid paralysis. That herpes simplex virus, well, rather than targeting motor neurons, that's going to target neurons in the temporal lobes, also the frontal lobes. So we're going to target emotional in memory and language areas. So we're going to see profound memory deficits and language deficits with herpes simplex virus infection. In both cases, we're infecting and killing off neurons, but again, location is key here. Okay, aside from viruses, we got other options. Prions are pretty unique. Prions, we see an example here, and I'm going to butcher this name, I'm sure, but that's not new for me. I believe it's Creutzfeldt-Jakob disease. This is a prion disease that causes progressive neurological symptoms. So the patients are going to eventually be paralyzed and only have these kind of involuntary jerks. They have hallucinations, cognitive memory impairments, and within about a year or so are dead. So prion diseases have invariably poor prognoses. Now, a prion is just an infectious protein. You acquire these infectious proteins by eating infected meat. So we've probably heard of mad cow disease, things like that. So mad cow disease is a prion disease. Because of that, we wouldn't want to eat any of the cows who have mad cow disease. Now, that said, prions are somewhat species specific, so you might be okay. You certainly wouldn't want to eat infected human meat. That would guarantee prion disease for you. And this is one of the ways that we discovered them. I won't go into it here, but a bit of cannibalism led to discovery of prions in humans. On occasion, prions do jump ship though, so probably not a good idea to eat brain. I know brain sandwiches are all the rave at the fall festival, but brain tissue has a whole heck of a lot of prion protein. And if one of those prions misfolds, that's all it takes to cause a prion disease. As far as treatments go, we got nothing for you. So you don't want to get prion disease. You're going to die within a year. There's really nothing we can do. So what's prion protein doing? Well, normally, I don't know. Not much. You can knock out prion protein. When it's folded up, normally we call it cellular prion protein. So prion protein can really form two types of folding states. C, cellular, that's normal. What does it do? I don't know. Might have some subtle roles in learning and memory, but you can knock it out and mice live just fine. We can't really tell anything's wrong with them. Same protein can misfold, and that's all it is. It's not a mutation, it's a misfolding. The exact same amino acid structure, but folded up in a different way. Now it'll kill you. Structure is function. The exact same amino acid sequences with one structure, just fine. Another structure, kill you in a year. Why? Well, the way that it folds up leaves an awful lot of hydrophobic residues exposed. And there's your problem right there. A whole lot of hydrophobic amino acids are exposed. This causes two things. It causes it to stick to itself. So you get these little protein aggregates. It also causes the unfolded protein response. Because the thing about prion protein is that it's remarkably stable. You can eat it, run it through your digestive system, it'll still kill you. It's going to be the exact same structure by the time it comes out of your gut and into the bloodstream. So this misfolded, or apparently misfolded state is very stable, so there's nothing we can do to fix it. So prion protein enters a cell, the cell sees it as a misfolded protein, tries to refold it, not a chance of doing that. We can't misfold it. What's the responsible thing to do? Kill yourself. So that's what the unfolded protein response does. Triggers apoptosis. We got misfolded proteins, man. Let's call it so that we don't spread this problem to our neighbors. The problem is that prion protein is very stable. And it then causes the normal prion protein to misfold. So once we have the infectious prion protein in our system, it takes your normal prion protein and turns it into the infectious version. So this spreads like a disease, because it is one, throughout the body, causing the death of cells along the way via the unfolded protein response. And when we look at the brain tissue post-mortem, it's got a whole bunch of holes in it. If you look at this figure over here, on top, that's prion, infection, bottom, normal. Look at the pink one, the third column there. That's brain tissue, and you'll notice a whole bunch of holes on top. Because what the prion does is kill off brain tissue by triggering an unfolded protein response. Nasty stuff. Certainly not worth the slight bit of exhilaration you get by eating a nasty-ass brain sandwich. The last pathogen we'll touch on here is the brain-eating amoeba. It's exactly what it sounds like. It's an amoeba that eats your brain. The brain-eating amoeba very rarely infects people. It's only a handful of cases per year, but when it does, it's almost always fatal. So it's worth knowing where these live. They live in all freshwater lakes in the southern United States where it's nice and warm. If you get a little bit of lake water with the amoeba in it into your nose, it can cross that cribriform plate there and enter the central nervous system. And what it will do is crawl along, poking its flagellum into a nerve cell here and a nerve cell there, popping them open and eating the cytoplasm. Pretty nasty stuff. There's only a few survivors. Of course, they have some lifelong problems after surviving this, but most folks die. The diagnosis is confirmed by CSF samples and finding the amoeba floating around in there. So there's your gold standard there. All right, that wraps up our discussion of infectious diseases. If you have any questions, fill out the questions box. I'll see you later.